exciting to introduce this PopBio on the Dark Side seminar. So my name is Giulia Ghedini and I'm a lecturer at Monash University. And the PopBio on the Dark Side is an initiative for who's new, led by Courtney Taylor from Melbourne Uni. And the idea was to try to have a sort of regular seminar series centered on population biology and all things below and above, I guess, individuals and communities as well, but sort of centered more in like Asia Pacific time zones because of the struggles of for us connecting to seminar series that are happening in the US and in Europe. And yeah, so this is the 8th seminar, so we're still relatively new. And there's going to be one more seminar in November before the Christmas break. So normally it happens on the last Thursday of each month. And today is a bit of a special one because it's a joint seminar with the Response Diversity Network. And so I'll let Sam introduce the other slide.
Thanks, Julia. So yeah, hi everyone. For those who don't know me, I'm Sam Ross. I'm a community ecologist working in Okinawa in Japan. So also in the dark side, as it were. And so, yeah, I represent the steering committee of the Response Diversity Network, which is a bunch of folks interested in ecological stability and its drivers and these kinds of things, and particularly the idea of response diversity that I'm not going to get into a lot of detail here. But yeah, we also have a monthly seminar series that runs on the last Wednesday of every month. And by pure coincidence, Meghna was due to give a talk both yesterday and today, and we thought that seems kind of crazy, so we should just join forces. So we have. So Meghna's gonna talk today instead. So we also run these seminars every month. This will be our 18th, I think, so a little bit less young than the population on the dark side, and. Yeah, we also upload to YouTube. What I should say is we also record the Q&A. So if you have a question and you do not want to appear on video, please write your question in the chat and we can read the question to Meghna at the end of her talk. So now I will introduce Meghna Krishnadas, who is at the National Center for Biological Sciences in Bangalore in India. And she runs the Community and Functional Ecology Lab. And yeah, I'm looking forward to hearing what you have to say today, Meghna. Lots of stuff about traits and demographic rates and things like this, I think. So whenever you're ready.
The trait stuff isn't there so much, but maybe next time.
Sounds good. Yeah.
Just a flavor. All right. Thank you, Sam. Thank you, Julia. Just give me a second while I share my screen to start the talk. Could you confirm if you can see my screen?
Yep, there we go.
Okay, great. All right. Well, again, thank you, Julia. Thank you, Sam, for that introduction and for both the networks to give me this opportunity to present my work here. I'm going to talk today about the demographic response of plants to abiotic and biotic drivers, both individually and in combination. And in a bit, I will lay out the broader context which motivates some of this work. But I was watching some of the talks in both of these seminar series, and I realized that my talk today is going to have a different flavor from what has happened so far, but perhaps broadly connected conceptually to the discussions that have been ongoing. So, for example, at the PopBio series, I noticed that many of the talks, initial talks, were more on the theoretical ecology side. They were really interesting, but our lab is a very hardcore field biology, data-driven, empirical lab, and so that's kind of what we do. We work in out there in the forest, in these natural ecosystems, generating some of the data that can perhaps be used to confront these theoretical models, which are talking about how ecosystems or communities respond and might change in response to different drivers. And when I was looking at the Response Diversity Network, of course, as expected, most of the talks there are more about community relating community properties and diversity in particular to stability. And again, that is work that we are sort of starting in the lab. We haven't gone into that yet, mainly because of the difficulty of doing things like that in the kind of systems I work in, which are long-lived trees in tropical, fairly diverse tropical rainforests. But we have initiated some new work, which hopefully in the future I'll be able to share, which is in montane grasslands, and we might be getting into some of those diversity stability aspects. But on to what we will be talking about today. Can you see my slide change? Just confirming. Okay,
Yeah, that's
great.
great.
So I think for most of us here, we are aware that ecosystems around the world are today subject to fairly dramatic changes due to many human-mediated activities, which have led to habitat loss and fragmentation, nutrient deposition due to runoff from agricultural systems, and increased fertilization, use of fertilizers, the advent or incursion of invasive species into many natural ecosystems, and of course, the looming specter of climate change that we know is likely to have increasing implications as time goes by. And many of these changes, these global environmental changes, either individually or in cohorts sometimes, can disrupt and is now being seen to disrupt ecological communities. Now, to understand the disassembly and perhaps reassembly of ecological communities in response to these ongoing and predicted disturbances, today more than ever, perhaps we need to appreciate and understand the fundamentals of how ecological communities assemble, what determines their structure in terms of species relative abundances, commonness and rarity of species, and eventually how diversity is maintained in many of these systems and how the processes that maintain these patterns of diversity might change in response to the to global environmental changes that we are seeing around us. Now, the patterns of assembly, structure, diversity emerge from fundamentally from the way organisms respond to their biotic and abiotic environment. It's just baseline, basic ecological knowledge that not all species are everywhere, and that is because they differ in their ability to tolerate different conditions, which determines where they can be found in the first place, where they can persist in the first place. But wherever they are able to persist, they also have to compete with other species. They have a biotic environment they need to deal with, interact with other organisms, and these biotic interactions can then shape the eventual realized distributions of organisms that we see around the planet. And because of the differences in organismal response to biotic and abiotic drivers, and because of their eventual realized distributions, we also find that diversity is not uniformly distributed in geographic space. One of the best known examples of such spatial variation in diversity comes from the latitudinal diversity gradient, where for many taxa, and here illustrated for trees, we find that the tropical regions tend to be much more species rich. For example, the species richness of trees in 1 hectare across the tropical regions in this figure is much higher, and this declines as we go towards the temperate regions. And this latitudinal diversity gradient has fascinated ecologists for since perhaps the dawn of ecology. And there have been a lot of efforts to understand what generates these diversity gradients, which is partly or primarily perhaps historical and biogeographic, with tropical areas having had longer time for evolution to take place, and in relatively more historically stable climates, which might have led to the development of stronger specialization in these species, which could be in terms of finer partitioning of resources, or the development of strong biotic interactions. And by this, I mean host-specific interactions, both of which together or individually can lead to greater niche differences among species. And this niche difference can prevent species exclusion to then help maintain the high diversity that we see in the tropics. So which means that this question of why are the tropics still so diverse? How do they remain so diverse? And this diversity is not decaying might perhaps be related to the fact that we might see stronger biotic interactions and a stronger stabilization of populations in the tropics. And the background to this, of course, is in the fundamentals of coexistence, which is that a basic tenet or a necessary condition for coexistence is that competition should be stronger within species than among species. And when this happens, species will self-limit in that And this could happen because of differences in the way they use resources, in the host specificity of pathogens, or how they partition time and space. But regardless of the mechanism, the outcome is that increased self-limitation of species can stabilize populations against extinction, local extinction. That is, it allows them to recover when rare and allows them to, buffers them against exclusion from the community, but also might prevent an abundant species from growing uncontrollably, and their population growth slows down population growth when species become abundant, thereby allowing rare species to persist in the community. So this begs the question that when we have these geographical gradients in diversity, like the latitudinal gradient, should we see stronger self-limitation in areas of higher diversity? Because one would surmise, I'm not 100% certain about this and convinced about this, but one would surmise that for many rare species to persist, you have to have a strong degree of self-limitation. And this is not required as much, or this may not be as strong when we move to low diversity systems. Now, in many systems, particularly long-lived tree communities that we are interested in, studying self-limitation or recording self-limitation in terms of population growth rates can be intractably hard. It can take years and years of data. The data can be noisy, and it is very difficult to really get at these signals in perhaps a theoretically most robust way. Right? But we can pick up some signals of self-limitation from data that is more tractable. And one way to do this is by looking at the performance, demographic performance of individuals of different species in neighborhoods where they are surrounded by their own species and other species. So imagine in this hypothetical neighborhood of 2 trees, or a hypothetical forest of 2 trees, you can have a neighborhood with a target tree here where it is it interacts, but either directly for resources or indirectly via pathogens or mutualists with members of its own species and other species. And you can see how different individuals of a species, when you draw circles to define its neighborhood around it, can experience different densities of their conspecifics and heterospecifics. Now, the idea is that can we check, or we can check whether the demographic performance of the individual of a species changes as the densities of conspecifics and heterospecifics increase. Now, from this, we can get a signal, is the impact of conspecifics on demographic performance more negative than that of heterospecifics? And if that is the case, then we can interpret that as a signal of stabilizing self-limitation. That is, intraspecific competition is likely to be stronger than interspecific competition in the system, which might translate to a stabilizing effect on population dynamics, thereby leading or contributing to the maintenance of diversity in such systems. Right? And so then, people have thought of this before. Of course, it's not, a completely new question. We've been trying to grapple with this for some time, like I said. And a recent study by Lisa Hulsman, which was published in Nature very recently, checked whether the signal of stabilizing self-limitation, which is conspecific effects being more negative than heterospecific effects on average, is it stronger in lower latitudes? And they found no signal across the latitude. Right? Now, this was intriguing, but one reason why this might be the case is because studies from single sites show that conspecific density dependence, negative or otherwise, and stabilizing signals are probably the strongest during early life stages of many trees in many forests. Right? And the Hulsman study only dealt with larger size classes, and many of the studies previously also have only dealt with larger size classes. So what we thought is maybe we could compare how the strength of stabilizing self-limitation changes or varies across the latitude at different size classes. So we put together data from 21 forest plots across the world. This is part of the network of the Forest Geo Network and the Chinese Academy of Sciences, their network of plots. And we modeled how individual survival at different life stages of seedling, sapling, and trees change in response to increasing density of conspecific and heterospecific neighbors. We took the difference between them, which is what we term stabilizing self-limitation, and checked how that varied with a gradient in latitude. And interestingly, we find that just as Lisa Hulsman did, in stabilizing self-limitation for sapling and trees, or the older life stages, across the latitudinal gradient. But for seedlings, we do see a signal that lower latitudes seem to have a stronger negative effects of conspecifics relative to heterospecifics for seedling population, for seedling demographic performance. Now, latitude, while the latitudinal diversity gradient is fascinating, latitude eventually is just a line on the map that we have drawn to represent certain changes in what are likely to be the mechanistic drivers of ecological pattern and process, right? And what really changes across the latitude is the climate. Mean annual precipitation and temperature are two major drivers that change, but by and large, moisture or moisture availability, which we characterized here as an aridity index, changes quite drastically, taking into account both temperature, precipitation, and their seasonality. It changes quite drastically from lower latitudes to temperate and other higher latitudes, right? So what we thought is, okay, is this change, or is the pattern of latitudinal diversity gradient, self-limitation with latitude related to the change in the abiotic climatic conditions that we see around here, right? And yes, when we ran a mediation model where we checked whether the trends with latitude in negative density dependence were actually mediated by climate, we found that of all the climate variables, moisture availability was a strong and very prominent predictor of the difference in stabilizing self-limitation. And in fact, when we had moisture in the model, the effect of latitude per se disappeared completely, suggesting that the trends that we're seeing with latitude is almost entirely driven by the fact that moisture availability changes across a gradient, which also suggests a generality that it is not just about the latitude, but moisture or differences in moisture availability, keeping all else equal, is likely to drive these patterns of stabilizing processes or the strength of stabilizing processes that we may see across geographic space. Now, just for consistency, we also wanted to see whether, with consistency with other studies, we wanted to see whether the strength of self-limitation also changes with species abundances. Because, one of the interpretations from Lisa Hulsman's work was that even though there was no latitudinal gradient, rare species, the really rare species in the tropics were experienced stronger self-limitation in tropical climates. Now, we saw that for seedling, this was not the case at all. We saw no signal with respect to latitude and any interaction between latitude and abundance. But again, with respect to moisture availability, we saw that in more moist sites, wherever there was, wetter, more aseasonal sites, the rarer species tended to experience much stronger density dependence. But I have to say here that this was really driven by one tropical site, or largely governed by one tropical site, which is Yasuni, which had many, many rare species. And which makes me think that we're not very sure if this is really a standard pattern that we see across, or is it more a case of, historical factors determining, the diversity begetting more diversity and stronger, more specialized interactions, which might then lead to some rare species experiencing this sort of more negative impact in these wetter, more stable climates. But overall, we found that stabilizing self-limitation was primarily driven by climate, moisture availability specifically, and it is likely to regulate community structure in these wetter sites, and that may arise from finer niche partitioning due to biotic interactions, such as between plants and pathogens, which experiments have shown are one of the key drivers of density-dependent mortality during early life stages. And resource partitioning is unlikely to be explaining something like this in plants, right? But which begs the question that we need to understand the underlying processes that might lead to these gradients in stabilizing self-limitation. But with respect to its link to diversity itself, we found that, as expected, rarefied richness, a measure of diversity in these large forest plots, was positively correlated with moisture availability, and also positively or correlated with strength of stabilizing self-limitation. That is, high diverse sites had stronger self-limitation. But again, when we put this together in a mediation model, it seemed like while these direct correlations were there, stabilizing self-limitation did not appear to be the main mediator of the latitudinal pattern in diversity that we see. So that still, leaves us with the question, like, is this a cause or an effect of diversity which has arisen from more historical and biogeographic reasons that we see today. And so it would be interesting, I think, to see if there are ways that we can tease those apart. Like, is this stabilizing self-limitation? It might be contributing to maintaining diversity, but how did it come about in the first place? And how does it vary from, why does it vary from site to site is something that I think still needs deeper investigation. But this relationship between self-limitation and moisture is something that we are seeing more and more. And in a recent study by Edwin Lebria-Treos, which used 20 years of data on seedling demographic performance in Barro Colorado Island, they found that seedling negative density dependence becomes more negative in wetter years when the soil moisture increases. And they used a bunch of simulations to show that this also changes or explains patterns of local seedling diversity from between wet and dry years, where stronger CNDD corresponded with the maintenance of higher local diversity in seedling assemblages during the wetter years, right? So there's an interesting finding, of course, and that got us thinking, like, is there most of these studies that look at the density-dependent interactions often look at it or forget about the background abiotic conditions in which this plays out. But we know that in many systems, abiotic conditions can be quite heterogeneous at small spatial scales as well. Now, what does that mean for density-dependent interactions in such systems? And that's something that we wanted to find out. Again, we focused on seedlings and seedling dynamics or seedling demographic performance to look at this, and conducted this study in a human-modified forest in the Western Ghats. And the reason we picked the system, well, one is because permits in India are really hard, and a private forest like this allows us to do experimental and long-term manipulations without worrying about too many permits. But also, ecologically speaking, this system generates these gradients spatial gradients in abiotic conditions, and allows us to test whether biotic processes show context dependence with respect to these abiotic gradients. And we set up a series of plots across this landscape, about 1,000 plots, in which we monitored seedling demographic performance. We tagged individuals, we recorded their survival and growth, and now we've been monitoring it for about 4 or 5 years. This is the 5th year that we are monitoring it, and also measured the primarily light, water, and soil nutrient conditions to see if the strength of density dependence varied with the change or a gradient in these conditions. And I'm sorry about this messy figure, but let me lead you through it. I thought it might be important to see all of these. What we find for the species for which we had sufficient data to run some of these very complex and data-heavy models is that most species that had higher survival at low conspecific densities, or when they were just alone, surrounded by no conspecifics, they also maintained fairly high survival even when conspecifics increased, right? So as a result of this, many species actually did not show a response to conspecific neighbors. And this is, by the way, this is conspecific effects when accounting for heterospecific effects, right? So it's stabilizing conspecific effects potentially. But this is only with respect to seedling neighbors. And we went with seedling neighbors, because of resource and personnel limitations. We are working in fairly low resource conditions here in these systems, but also because studies like Edwin Lebria-Treos and previously from BCI, they show that larger individuals might have an effect, but it is the seedling neighborhood which seems to have the strongest density-dependent effect on individual seedling performance. But anyway, what we find overall is that the effect of the change from baseline survival when you add a mean conspecific density that is experienced by individuals is prominent only for 5 or 6 species out of these 40 species that we analyzed, right? And the negative effect of conspecific seems to manifest only in wetter conditions, that is when soil moisture is high. And in drier conditions, or the other way to look at it is where the conditions become drier, the stabilizing effect of density dependence, particularly on what seem to be the abundant species in these systems, which by the numbers of the mean conspecific densities that they experience, seems to be prominent or a sort of community compensatory trend seems to be prominent only when conditions are wetter. And in drier conditions, these species or the species that are abundant are likely to have lesser control or lesser negative impacts on their seedling emergence and seedling abundances as compared to when the conditions are better. Now, this is, of course, a concern because we are looking at these human modified systems where parts of these systems or large parts of the systems due to edge effects become much warmer and drier. But we also found another interesting phenomenon, which is that because we used GAMs, which could model the nonlinear responses of seedlings to their neighbors, we found that at high conspecific densities, interestingly, a few of these abundant species tended to have positive response to conspecifics. And this is something that we've been wondering why it is happening. It could be because in drier conditions, these species might harness more mycorrhizae. And this has been seen in some systems. We don't know whether this is the case from this system, and that's something we want to look into in the future. But also it appears that you need to have sufficiently high densities to even benefit from these positive interactions, which is something we've been wondering about. Why is it that, this is happening? Is it something to do with the critical mass or critical density that then allows you to have these facilitative or more positive interactions with your neighbors, right? But one thing that is missing, of course, is the impact of large trees. And this is something that we are getting into now. So perhaps at a future date, we will be looking into whether these patterns of context dependence in conspecific effects that we see are also or equally similarly mediated by the larger trees in the neighborhood. But for now, this is what we have. And we also see that this shift from positive to negative conspecific densities can be explained by some commonly measured species trait. And in this case, we used SLA and LDMC. I present the results here for SLA, where we find that the most important thing is that when soil moisture dips, we find that high SLA species are the ones that seem to enjoy this positive effect of conspecific. So these are perhaps the species that we should be looking for in terms of harnessing mycorrhizae, or looking at the mechanisms underlying this shift from negative to positive density-dependent performance when the abiotic context changes spatially, right? And we need to be reading some of these biotic interactions alongside the basic response of these species to the abiotic conditions that we see. So we've now been tracking about over 12,000 seedlings, and we've used this information to just understand how seedling survival responds to the moisture and nutrient gradients, like multiple abiotic gradients that we see around in the landscape. And we find that moisture again comes up as one of the top drivers or a key driver of seedling performance. But also we see that there's not much diversity in the responses of species. By and large, nearly all species show the same trend as the mean trend that is estimated in the community, which is that where there is higher moisture, these species or all of these species show better survival. And we don't really see a shift in the ranks or the competitive ranks of species as moisture changes, right? And this is not surprising, perhaps, because these are all species that are in a tropical, humid forest, evolved to withstand or, in fairly high moisture conditions. And you would think that within the moisture range that we studied, they are, most of them would perhaps, that this is not enough. The range that we have here right now is not enough, perhaps, to shift the competitive ranks of species. But we are wondering whether this means that moisture may not really alter, like even when moisture dips really low, and that has been happening in the recent past, where soil moisture levels have been going really much lower than we've known them to do before. Unfortunately, there is little baseline data for us to go by, and our data is one of the first for the system to even characterize these changes. But what we find is that fitness differences, perhaps, or what we are thinking is that fitness differences between species might not be altered so much by the changes in these abiotic conditions. And that should make us think even more about what the shifts in biotic interactions, such as stabilizing density dependence, mean for the maintenance of community structure in the system as we go into a future that is potentially warmer and drier and is seeing a lot of changes. And I would also like to here just point to another interesting finding that we got, which was unexpected, which is that phosphorus seems to have a very strong impact on seedling survival. And contrary to what we expected, it is available phosphorus in the soil. As it increases, you actually have poorer performance or demographic performance of seedlings. And again, by and large, this seems to be a fairly consistent trend across the community, but we do see that some species respond differently to phosphorus. And when you're thinking about it, it seems like, many tropical forest species they've evolved in phosphorus limited conditions. And when you have a high input of phosphorus, as might happen in this system, which is close to an agricultural landscape, which is, this is forest fragments interspersed within where there is fertilizer application. But it is representative of tropical systems or tropical forests in many human modified landscapes, where you have forests abutting agriculture and there's a huge nutrient runoff. And I think we need to be looking at the potential impact of nutrient addition on forest regeneration a little bit more. In this regard, we have looked at traits, and we think that some of the relationship between survival and phosphorus might actually be driven by the way phosphorus is implicated in xylem and stomatal behavior and how that regulates response to moisture. And that's something that we hope to be looking into in the future. But overall, we do see signals that there is context dependence in density-dependent performance, which is likely to emerge from the biotic interactions that mediate density dependence. This has potential implications for both spatial storage effects, but also temporally, if you have better years versus dry years, like Edwin's study showed. What does that mean with respect to which species are doing better? And at least from the data that we have so far, it suggests that we're not really seeing a storage effect kind of. We haven't done the rigorous analysis for this, but we are not really seeing what might be a storage effect where sites where species are doing better, they also have stronger density-dependent mortality. And so we might, overall, what we think is that this sort of context dependence might play into the future of diversity or plant diversity, maintaining diversity in these human modified systems, and may drive how the community responds to changes in the climate, especially if there are parts of this forest which are likely to get warmer and drier. And we are also thinking, might this play into, or might this factor into our decisions for what species to choose for restoration in what kind of conditions? But of course, that's a translational component that's a bit ahead in the future. But some of these, the information that we have for seedlings do match up with some of a previous study that we had done on seed to seedling transition and how that changes with distance from the forest edge. And at that time, we had just used distance from edge as a proxy for changes in these variables. And basically, we used the baseline seed to seedling transition differences between edge and interior and changes in density dependent seed to seedling transition to fit these models where we looked at how species richness changes in these fragmented landscapes where you have landscapes that are just intact forest, landscape comprising of fragments with an interior patch, which is this black line. And if you have the same amount of forest, but the entire forest just feels behaves like an edge habitat, which are these red lines. And overall, we find that whether we look at the level of individual patches or the landscape as a whole, we see that landscapes that are fragmented have lower diversity than intact forest landscapes. And this dips even more drastically if there is no interior condition that sort of maintains these stabilizing effects that we see during key demographic transitions. And this pattern sort of holds even when we increase fragment size and we overall increase the area. As expected, larger patch sizes will have more diversity, but you still see an effect of fragmentation, habitat fragmentation playing up. And we see this with respect to richness and more clearly perhaps with respect to diversity as measured using inverse Simpson as well, suggesting that this sort of speaks to this debate about habitat amount versus habitat quality. And of course, when we look at habitat quality for multiple species, it's really hard. What does that even mean? But perhaps if we look at these biotic interactions that are such as stabilizing density dependence and how that operates in some of these, for some of these communities in fragmented landscapes, we might get a measure of habitat quality at a community level and be able to predict the future of diversity. And we use some of this information to also to look at why is it that diversity is not maintained in fragmented versus intact forests and with and without interior habitat. And we find and we used invader growth rates to derive some of these insights. And we found that in intact forest, pretty much all species had positive invader growth rates, and they were unlikely to go extinct. And extinction is characterized by these, you can have invader growth rate dip below 1, or you can have, there can be stochastic extinctions, as illustrated here by the open circles as opposed to the solid lines, which show that the species remains in the system. And then when we compare an intact forest to a fragment, which has an edge and an interior, the sort of realistic representation of fragments, we find that more species start to blink out of the system as time progresses, but even more so if the fragment becomes one without an interior habitat where these stabilizing biotic interactions are still operating. So then the next step, of course, is to understand, how is it that these biotic interactions themselves, which might be the mechanistic drivers of the demographic performance that we're seeing, how do they change in some of these systems of interest? And something that we've been going into a lot in the last couple of years is understanding plant-soil microbe interactions. And there's a very nice body of theory that has developed around the impact of microbes on plant species coexistence, particularly pairwise coexistence. And the basic premise here being that two plant species may cultivate the soil for their own unique set of microbes that can affect the cultivating species differently from the other or the heterospecific. And this can lead to either competitive differences between these plants, but also lead to a stabilizing effect that allows or promotes coexistence between these. And it's the relative impact of the fitness differences and the competitive effects that eventually decide whether a pair of plant species might coexist due to microbes. And these microbially mediated feedbacks can respond, or because microbes respond directly to abiotic conditions and as do plants. So these microbially mediated fitness differences and feedback can also change with abiotic context. And this is something that we have tested. This is just data for one pair of species where we set up an experiment with high water in combination of high and low water and high and low light, which is what these acronyms here suggest. HW is high water, LL is low light, then there's high light and low light, and so you can read the rest. And what we find is, and I'd like you to look here first at the sterile soil, which is just, if there were no microbes, what would be the coexistence outcome based on the fitness difference and the strength of stabilization between these species. And we find that microbes in high water, low light conditions, microbes are actually stabilizing and they are promoting coexistence by and large. But this shifts as we go towards low water, high light conditions, which sort of exemplify some of these edge conditions or future climatic conditions, where we find that one species enjoys a higher fitness difference and is likely to exclude the other species from their system. We also try to use this and the data from the density dependence models that we have to project future dynamics or longer term coexistence outcomes from these studies. And we find that the results are kind of not matching exactly what the predictions from the coexistence predictions from our experiments should be. And we find that some of the outcomes can also be driven by the fecundity of species. So if we just consider one life stage, that may not be sufficient for us to project longer term dynamics. And of course, this is a problem in many of these long-lived systems, right? But when we look at, if we are looking at equal fecundity of the two competing species here, we find that regardless of which condition it is in, it appears that future tree abundance, is higher for this one species in blue, which is Litsia, which is actually the opposite of what we found in the experiments, the seedling experiments that we did. However, if we account for fecundity differences, we find that in forest interior conditions, the two species are more likely to coexist, whereas one outcompetes the other as predicted by our experiments at forest edges where the abiotic condition changes, right? And so, of course, we did a whole lot of, we're still doing a whole lot of checks on this, but by and large, this seems to be the outcome that we see from these simulations using the empirical data that we have generated. But overall, I think the goal here is that, can we predict something about the future composition of seedling or the seedling bank by conducting these large-scale observations and experiments, maybe from the forest edge to interior, which gives us a very good idea of how abiotic conditions might change in the future as well, or stands as a proxy for temporal changes in abiotic condition as well. And to do so, we are using a variety of approaches, and traits are a big part of what we do in this lab. So we have measured, for many of these species, we have measured a whole bunch of traits. In fact, we did this massive trait campaign where we measured 21 different traits to characterize the whole plant phenotype, above ground, below ground, chemistry versus physiology. And now the goal is to relate this to performance across these gradients and across biotic interactions as well. And just to give you a flavor or leave you with a flavor of what we are up to, we are also planning to combine a lot of these experiments on plant soil microbe interactions with field experiments and monitoring to better understand some of these specific empirical drivers of what might be leading to these phenomenological outcomes of density dependence and plant, competitive differences in different abiotic and biotic conditions, right? In order to hopefully predict how diversity might respond to the changes that we're going to see in the future. And particularly when we have these human modified systems that are also subject to climate change, do we see an interaction between these factors in terms of how the community is structured and where it is likely to be headed in the future? And that, in effect, is what we are sort of doing in the Community and Functional Ecology Lab. Like I said, we are a completely field-based outfit here. Most of our work is solidly empirical, based on ground data. But I'm always happy to discuss more with theoretical ecologists how we can use some of this data to either confront theoretical models or have a theory data conversation. This is what we have so far. And with that, thank you for your attention, and I'm happy to take questions.
Thanks, Meghna. That was really fantastic. Lots of really interesting stuff there. Questions from the Zoom audience. A reminder that we are recording the Q&A, so if you don't want your voice or face to appear on the video, you can write in the chat and we can read, Meghna, your questions instead.
Should I stop sharing the screen, or do you think people might want to go to a particular slide? Yeah?
Yeah, maybe you can reshare if somebody asks for a particular thing, but yeah, maybe for now.
Sounds good.
Thanks.
Yeah.
And then I can better see who's here anyway.
While we let the audience warm up, was there a question?
Not yet.
No, okay. I will ask one because I have one. Maybe you mentioned something like in the interesting pattern you saw where there is a stronger density dependence from intraspecific competitors when there is more moisture. Maybe I missed it, but I think you mentioned something that maybe has to do with disease. Is that why you think what is happening?
Yeah, so disease, in the sense, we think that might be one of the drivers. We have seen some evidence for that in the seed to seedling transition experiments where we had sprayed fungicides and insecticides, and we found that the impact of fungi on density dependence is weaker at forest edges. And we do think, and the plant soil feedback experiments seem to suggest something similar, that soil microbes might be a driver, a mechanistic driver of some of these changes. But there is also, and it's kind of commonsensical, right? Like, in wetter conditions, some pathogens like mycetes are likely to spread more, they're more virulent, infection is stronger, infectivity is stronger. So we think that microbes might be a key driver of some of these patterns that we are seeing.
Okay, and in the system, so microbes are very species specific, I imagine?
Yeah, we don't know that. So we're just starting to develop some of that, or generate some of that data. We're doing these next-gen sequencing, and we're looking at microbes both in the roots of seedlings because it's easier, and also associated with testing for signals of whether the Janssen-Connell effect is prominent in tree species. So we're looking at microbes close to the tree versus away from the tree and seeing if there's a signal of host specificity. In fungi, we do find that there is both fungi and bacteria, we do find a signal of host specificity in the soil tree, where we find that we differ in the position that they harbor. This sort of stays even when the species, whether it's edge or interior. So something is happening with microbes, and we are now getting into the more the weeds of the detailed data for linking microbes to seedling performance. And we still don't have the data on seedlings, but we expect something very similar to happen.
I see we have a question in the chat. Sam, do you want to read it?
Oh, yeah, I can read it out. So this is from Xue Wei, and they ask, you've mentioned there's a negative effect between phosphorus and seedlings survival. Do you also find any evidence that soil nutrient conditions might affect the strength of CNDD?
That's a great question, and that's something that we are getting into now. The relationship with phosphorus, as I mentioned, was very unexpected. We had it there primarily as, just to account for the variability in nutrients when we are looking at light and water effects. It's something that we're going to check now, and depending on what we find, we might do some experiments to delve a bit deeper into it. The results that I presented for just seedling response to phosphorus, literally we got them like 2 days ago. So it's quite new.
Hot off the press. Great.
Yeah.
Yeah. Fantastic. Thanks. Yeah. Any more questions from the audience? You can write them in the chat or just put up your hand or just unmute yourself. While we wait, I have a very naive. Oh, go on, Dave. Go on.
Oh, Dave. Yeah. Hi, Dave.
Meghna. Great to see you. Great talk. I was wondering, I love the work that you guys did on like fragmentation, and I think more needs to be done on sort of like the demographic consequences of fragmentation. And I wonder if you may have already done this or may not have, and I don't know enough about the species actually that you were studying, but like, so animal dispersal, right, would be, of course, important and differentially important between the fragments. And in my mind, that could then like presumably change sort of the spatial relationships between the trees in which like the negative density dependence is interacting. So it seems like there could be some interaction between like fruit dispersal, seed dispersal, and negative density dependence. Have you modeled that or looked at that at all?
No, we haven't. Well, so in the paper that we Simon Stump and I wrote together, we modeled the effects of dispersal, and we do find that if there is, as dispersal limitation increases, it's more likely that you're gonna, the intensification of diversity loss becomes more intense, right? So there is clearly an effect of dispersal. We also find other signals of that when we are using this plant soil feedback data to, do some simulations where we find that differences in fecundity, which could effectively translate to, how much, and we didn't measure fecundity as just seeds, number of seeds, but how many seedlings are there to start off with, which often can be a product of dispersal, right? Or an outcome of dispersal. We do find some signal that how many seeds come into a patch or how many seeds arrive at a system has an effect on eventual coexistence outcomes. But if you're talking about empirical data, no, we haven't gone there yet. And I'd love to brainstorm about what exactly to do, because a lot of the dispersal literature that I've been reading, I've been finding it hard to reconnect it to the sort of work that we're doing, like go from one to the other. But yeah, I'd love to talk, have a deeper chat about it.
Yeah, cool. Thanks.
Thanks. And yeah, that was a more eloquent version of the question I was about to ask. So yeah, there's oh yeah, there's some more comments in the chat here about Xuewei was saying they also found a negative relationship between phosphorus and seedling survival along the Panama Canal, but they haven't looked at nutrient-mediated CNDD yet. So interesting.
Oh, that's really nice. And wow, we should talk. I'd be really curious to know what you think of the mechanisms that are driving this.
Yeah,
but I
like
think I was reading this paper. I think Joe wrote a meta-analysis of nutrient addition experiments in tropical systems, and I think maybe we need some more studies or globally distributed studies looking at something like that. Okay, there's another question.
Yeah, there's one here that says from Chinte that says, "Thank you for your rich talk. My question is based on your results. Could you explain whether moisture affects microbes or whether it directly affects CNDD intensity? Because I suppose the below ground fungal composition will differ under different moisture levels."
Great question. Yeah, again, one of those things that requires a lot of detailed empirical data and something that we are getting into now. We are trying to characterize what the fungal composition is in for different plant species and whether that changes at the edge and the interior. And in future, I think we also want to look at whether it changes directly with moisture availability or not. Edge may not be such a great proxy for abiotic changes. To answer your question, whether moisture affects the microbes directly or whether it indirectly affects CNDD, I think it might be a bit of both, but I'm not sure that we have teased that apart yet. We do see some signs that the microbial composition changes between edge and interior, which might be driven by moisture, but we need to do those. It's a lot of next-gen sequencing. It's a lot of heavy-duty lab work. So we are only getting into some of that now.
Thanks, Meghna. Sounds really exciting, though. It looks like you're doing a lot of really cool stuff that's like in progress at the minute. All that trait stuff seems like a lot of work.
Yeah. It was a huge campaign. It was quite a lot to do, but and so we have this really detailed trait data for about 16 species, which is not a lot in terms of number, but I think we've really got a great representation of the different strategies that are possible in this system. Yeah, so we're looking forward to relating some of that to the demographic performance of seedlings.
Yeah, fantastic. Great. Time for maybe one or two more questions if people have.
Meghna, great talk.
Hi, Varun.
I have a question. Hi. I have a question about the model that followed after the plant cell feedback experiment. It seems that you showed that the fecundity actually has a very large effect. I think I may have lost it, but the model combines the experiment and something else and some other source of data, right? So can you talk a little bit more about that and like why you think there's this mismatch?
Yeah, sorry for the lack of clarity. I think I really zoomed through that part because I was afraid I was going to take too much time. But yeah, you're right that fecundity seemed to have a large effect, or almost seemed to flip which species would coexist in the system and how long they coexisted. The model was based on the outputs from the plant soil feedback experiment, which you're kind of familiar with, which my student did, and also from the seedling survival data that we have from the observational plot. So basically density dependent responses of seedlings to conspecifics and heterospecific. And he pulled out some of that data to specifically look at how these two seedlings or these two species responded to conspecifics and the other species. Those are the two data components that were used to parameterize the simulation.
So I would have imagined that in the equal fecundity case, that would have matched your experimental results, but it seems that wasn't the case. It was actually when you assume there's a slight asymmetry in fecundity. So that's the part that because the experiment predictions assume equal fecundity and all else equal, right? So why wasn't it when you assume equal fecundity where you actually have the best match?
No, we did have the best match in terms of coexistence outcome. Again, sorry, I think I just sped through it. There was a lot there.
Sorry,
and I just
my bad.
No, my bad. I apologize. But the equal fecundity scenario showed that the two species were likely to coexist for a long, but only for some time. Like, initially they would, and then despite the prediction from the experiment model, over time, it seems like one of the species would still go extinct. And it is a species that was predicted from one of the contexts in the experiment, right? But what did not match is that the patterns did not seem to change regardless of abiotic context, even though we found that abiotic context in the experiment altered coexistence outcomes, right? And I think part of that might be because you saw the data, there's like a lot of noise around that data. So we didn't get really clear patterns for these two species. And I think that's what might be just that margin of error. When we incorporate that error into the predictions, it's just leading to these longer term outcomes that might be different, or that are different from what we would interpret from just the short term presentation of the empirical data. And that completely switches. So the species that's expected to persist in the system, when if you change fecundity, that's the one that actually goes out.
Yeah, thanks a lot. Great talk.
But I'd love to talk to you more about this. I've been meaning to write to you and think that
Yeah,
maybe we should have a chat
whenever.
about it.
Yeah.
Sounds great. Thanks so much, Meghna. I think that's the hour, so we should probably wrap this up. Julia, is there anything else we need to say, or should we just thank Meghna again?
No, I think, we just need to thank Meghna for this exciting talk, and like really looking forward to the next time you're going to present your new research. Yeah. It was really cool.
Yeah,
Thank
really
you.
great.
Thank you so much. And thank you once again.
Yeah, awesome.
Great. So
Thanks,
thank you.
everyone.
Thank